Good morning and welcome back to botany class. In the previous class, we discussed the process of fertilization. This class too, we are going to discuss the events of fertilization. Define fertilization. Fertilization is the fusion of male and female gametes. When you see the structure of gynoecium, it contains stigma, style and ovary. In the stigmatic surface, the pollen grains are dusted. This pollen grain start to germinate. It develops the pollen tube. So today we start this event from pollen tube in the style. The pollen grain, when it dusted on the stigmatic surface, it starts to germinate. This is the pollen tube. This is the stigmatic surface. This is the style. This pollen tube grows towards the style, ovary, and inside the ovary, the ovule and embryo sac. It reaches the embryo sac and fuses with the egg and forms the zygote. That's the process fertilization. Before that, let's see different types of style. The pollen tube grows along with the stylar region. It depends upon the types of style. There are three different types of style named as hollow style, solid style and semi-solid style. There are three different types of style. Number one, hollow style. It's otherwise called open style. Solid style is otherwise called closed style. And the semi-solid style is otherwise called half closed type. So, three different types of style is observed. Among this, we are going to discuss in detail about the types of style. First type is hollow style. In this style is observed in monocots. In this type of style, there is a hollow canal. This is the stylar region. There is a hollow canal running from the stigmatic surface to the end of the style is present. It secretes a mucilaginous substance. Through this, the pollen tube grows. So, in this canal, there is a mucilaginous substance. The pollen tube grows along with this canal, and this mucilaginous substance, it nourishes this pollen tube. So, this stylar region which is filled with secretions. The secretions which contains carbohydrates, lipids and some enzymes like esterases, acid phosphatases, as well as it checks the incompatibility reaction between this pollen tube and this style, because when this pollen tube are germinating, it passes through this. So, this region is lined by this hollow canal. It's lined by a glandular tissue called transmitting tissue. Transmitting tissue. In this type of style, hollow style, this canal is lined with a single layer of glandular cells. Solid style. Second type of style is solid style. It's common among dicots. This type of style is common among monocots, but the second type of style is common among dicots. It's characterized with a central core of specialized, highly specialized cell called transmitting tissue. The transmitting tissue is the specialized characteristic feature of this solid style, which is otherwise called closed type of style. So, in this style, the transmitting tissue is present in the central core, and the function of this is similar to the function of the linear tissue, which is lining, single lining tissue, glandular tissue. So, it does the similar function of this type. And the pollen tube, in the first type, the pollen tube grows on the surface of the lining tissue. In this tissue, solid style, here is a central core of cell. So, it passes through the intercellular spaces. The pollen tube passes through the intercellular spaces of this style or region. Then semi-solid. In semi-solid type of style, it's the intermediate between the hollow style and the solid style. It's the intermediate. There are different opinions among the authors. Some of the authors, they suggest that their opinion is this transmitting tissue is present only in the solid style. But some others some other authors suggest the transmitting tissue also present in this hollow style also. So, first question you are going to learn is types of style. There are 3 different types of style observed: hollow type, solid and semi-solid. Now, second topic is entry of pollen tube into the ovule. Now, here is the stigma, style and this is ovary. This pollen tube germinates, it grows along the length of the, it travels along the length of the style and enter into the ovary. Inner to this ovary, ovule and embryo sac is there. So, it has to reach the pollen tube which reaches the embryo sac. Here is the embryo sac. So, it has to elongate. It elongates and enter inside. So, towards this, inner to this, there are synergids, antipodals and the polar nuclei is present. So, this tube, pollen tube, when it travels along the stylar region and the ovary and towards the micropylar region is due to the chemotropic substance. There is a chemotropic substance, due to this it leads to the entry of this. But when it moves towards the micropylar region, there is a structure called obturator. This obturator guides this pollen tube to enter through the micropylar region. So, this is the micropylar region, this is the chalazial region, and in the micropylar region the egg is present. So, fertilization takes place by this male gamete is carrying through this pollen tube. So, pollen tube which contains 2 male gametes, vegetative tube nucleus and the cytoplasm. All these are discharged into this embryo sac. The embryo sac consists of synergids. These all contents of the pollen tube which discharge into any one of the synergid. So, if it fuses to the egg, it is called syngamy. So, in this angiospermic fertilization which is called double fertilization. Double fertilization. What is meant by double fertilization? In the year 1898 and 1899, S.T. Navashin and L. Gainard, they observed double fertilization in Lilium and Fritillaria. In Lilium and Fritillaria, these two persons, Navashin and Gainard, observed double fertilization, which is common among in dicots. So, this double fertilization, there are two fusions taking place. What are they? There are 2 male gametes carried by the pollen tube. One fuses with the egg, another fuses with the polar nuclei. So, 2 times fusion taking place called double fertilization. So, let us see the process of this double fertilization. This is the pollen This is pollen tube. This pollen contains 2 male gametes. These are the male gametes. And it contains a tube nucleus, vegetative nucleus. So, pollen grain contains 2 male gamete and 1 vegetative nucleus. This is N in number. Vegetative nucleus is N. The male gamete also N in number, haploid. Then in the embryo sac, which contains synergids, antipodals and polar nuclei. This is embryo sac. This is pollen grain, and this is ovule. So, fusion taking place, double fertilization, how it takes place. This is antipodals. This is egg, these are the synergid, this is the polar nucleus. Sometimes this polar nuclei, 2 polar nuclei fuse together and forms as a secondary nucleus. So, now it's secondary nucleus. In double fertilization, this first male gamete, one of the male gamete, this N fuses with the egg. So, this egg cell also N. So, N plus N, it forms the zygote, formation of zygote. Now, the zygote is 2N, diploid, because male gamete is N and this egg cell is N, haploid. Both the haploid cells fuse together and it becomes diploid, 2N. So, zygote is 2N, diploid condition. And another male gamete, this male gamete fuses with, this is N, this N fuses with secondary nucleus. Here at first it was polar nuclei, 2 polar nuclei fuse together and forms as the secondary nucleus. So, this male gamete fuses with the secondary nucleus. So, this secondary nucleus already it is 2N in nature. So, this 2N plus 1N, now it becomes 3N. So, it forms endosperm. Now, this endosperm is 3N. So, from the zygote, the embryo develops. From this secondary nucleus, endosperm is developed. Understand this very clearly, this area, one word area. Here they may ask what is the number of zygote. Zygote is haploid or diploid or triploid. Endosperm is triploid, zygote is diploid, and this male gametes are haploid. So, n number. The options they may given and ask different types of question in one verse. So, understand it very clearly. And finally, it produces the endosperm, formation of endosperm. This is endosperm. Inner to this, there is a embryo. So, now this is embryo and this is the endosperm. So, this endosperm nourishes the developing embryo. So, what is the function of this endosperm? Endosperm nourishes the developing embryo. So, this fusion of first fusion, this male gamete fuses with the egg. The process is called as syngamy. Syngamy. 2 more question, very important. What is syngamy? Fusion of male gamete with egg. That is syngamy. Then now this fertilization is called as triple fusion, because one male gamete, this N, fuses with secondary nucleus 2N. So it becomes 3N in number. So since it involved 3 number of nucleus, it's called triploid. So here another question, what is meant by triploid? The definition, 2 more question, what is syngamy? And this fusion is triple fusion. The 2 process of this fertilization is double fertilization. The process or this phenomenon is called as double fertilization, and this fusion is named as triple fusion. And fusion of male gamete with egg is syngamy. It forms the zygote. So 2 more questions are very important: syngamy, double fertilization and triple fusion. From zygote, embryo is formed. From this secondary nucleus, endosperm is developed. After this, there are number of changes. That is, the next process is called as post fertilization changes. Post fertilization changes. There are number of changes observed after fertilization in the floral parts. You know, there are number of floral parts: calyx, corolla, androecium, gynoecium, where sepal, petal, this stigma, this stigma, style, this all parts are present in the flower. After fertilization, these all the non-essential word. This androecium, gynoecium, which comes under essential word, the petal, sepal and this this stigmatic region and the stylar region, this all wither and fall off. After fertilization, these all wither and fall. And this synergids, this synergid antipodals also disintegrate. And the micropylar region, this micropyle of ovule becomes the micropyle of seed to facilitate oxygen and water uptake. And this egg, the egg changes into zygote. Now egg, fusion of egg, after that the zygote, it forms the zygote, and the zygote develops into embryo, and the secondary nucleus forms the endosperm. And the ovary, this is the ovary becomes the fruit, and ovule becomes the seed. So after fertilization, what are the process, what are the events? Development of this embryo, development of this endosperm, and formation of fruit and seed. So this ovary become fruit, and this ovule become inside the ovule, which becomes seed. So this embryo developed inside the endosperm. So endosperm is the nutritive tissue of the developing embryo. Then depend upon the mode of development, there are 3 different types of endosperm. The endosperm is the result of triple fusion. The endosperm, the male gamete fuses with the secondary nucleus and forms primary endosperm nucleus, PEN, primary endosperm nucleus. From primary endosperm nucleus, endosperm is formed. So this primary endosperm nucleus is 3N in number, 3N chromosomes in it. Depend upon the mode of development, it is classified into 3 different types of endosperm. They are nuclear endosperm, cellular endosperm, and helobial endosperm. There are 3 different types of endosperm. How these endosperms are developed? This, as soon as this endosperm develops, the primary endosperm nucleus develops, it starts to divide before the zygote divides. Before the zygote divides, the endosperm starts to divide. So, endosperm undergo number of mitotic divisions and it is not followed by cell wall formation. See, this is the endosperm. Here, the cell primary endosperm nucleus first divides without cell wall formation. Here the cell without cell wall formation is called nuclear endosperm. So, the condition, free nuclear condition exists in the endosperm. This is the free nuclear condition which exists inside the endosperm is called nuclear endosperm. But in cellular endosperm, the primary endosperm nucleus divides followed by cell wall formation. So, subsequent divisions which followed by formation of cell wall. In the diagram, the cell wall formation taking place. Here is the cell wall, formation of cell wall. This is the second type, cellular endosperm. And helobial endosperm. In helobial endosperm, this primary endosperm nucleus moves towards the micropylar region, moves towards the basal region of the embryo sac. So, in this diagram, it moves towards the basal region of embryo sac and followed by developing two different chambers, a large chamber in the micropylar end and a small chamber in the chalacal end. This is the micropylar region, this is the chalacal region. So, this region is larger region where the cells not forms the cell wall. Cell wall formation does not take place and free nuclear division takes place in the micropylar region, and this is the chalacal region. It may or may not divide. So, it is observed in hydrilla and vallisneria. So, depend upon the mode of development, there are three different types of endosperm. Apart from this, one more endosperm is there, ruminate endosperm. In this, the endosperm formation is uneven or irregularity in its surface. So, it is called ruminate endosperm. What is the function of endosperm? The function of endosperm is the endosperm nourishes the developing embryo, and in most of the cases the endosperm divides before the division of the zygote. So, before the zygote divides, this endosperm divides. So, up to this you learn in this class all these questions. So, first question is start from types of style, 5 mark question, then post fertilization changes, then types of endosperm, and this one, this fusion, double fertilization and triple fusion. The questions you will get it through WhatsApp. Thank you.